Dear students, now we are going to see combinatorial synthesis. Here, large number of compounds are produced in a short period of time using a defined reaction route with a large variety of starting materials and reagents. Finally, the produced compounds are different but they are structurally similar. So, we can get a large number of molecules and that molecules are different but they are structurally similar. How they are produced in a short period of time using a defined reaction route. So, combinatorial synthesis is a technique in which large number of compounds can be produced in a short period of time using a defined reaction route and a large variety of starting materials and reagents. How the compounds are produced? They are different but they are structurally similar. So, we can get a mixture of different compounds in the same reaction vessel. So, what is the main condition? So, same in the same reaction condition, in same reaction vessel, we are getting large number of compounds. So, we can get more compounds in a short period of time. So, from this, we can get high chances of getting a lead compound. So what is combinatorial chemistry? This is an approach. So by means of this approach, we can synthesize a large number of molecules and that collection of molecule is called as a library. Then that libraries of compounds should be screened. Why that should be screened? To isolate a molecule having desired activity. So what is combinatorial chemistry? So here this is an approach for synthesizing the large number of molecules and the molecules should be collected and that collection is called as a library and the libraries of compounds should be screened to isolate the molecules of desired property. Now we'll see the stages of drug development and the role of the combinatorial synthesis. So in the case of drug development, first we are finding a target, then we are finding a lead. We have to isolate the active structure or active compound, then that structure would be identified. We are going for structure activity relationship and finally we have to optimize the lead. So these are the stages of drug development. Now this combinatorial synthesis is playing a major role in these three. One is finding a lead, structure activity relationship and optimizing a lead. Now we are comparing the conventional method with the combinatorial method. So in the case of conventional method, we are synthesizing only one molecule at a time. Whereas here we can synthesize many molecules or many compounds at a time. First we are making a compound. Then we are purifying that and we are going for testing the compound. Whereas here we are making this. Then we are testing. Finally we are going for purification. In a month, we can get hundreds of molecules, whereas here we can get thousands of molecules. Here the lead generation is slower, whereas here that is faster. Risk of failure is high, whereas here that is low. Now we'll see the principle behind combinatorial synthesis. So in the case of conventional reaction, we are taking two reagents. For example, that is A and B. We are combining that and finally we are getting a compound that is AB. Whereas in the case of combinatorial synthesis, we are taking n number of A with n number of B. They were combined and finally we are getting n number of A with n number of B. For example, A1, A2, up to AN we are taking, then B1, B2 up to BN we are taking. So this A1 is combining with these three. So how do we get that? A1, B1, then A1, B2, A1, BN. Similarly, A2 is reacting with these three and AN is reacting with these three. So A2, B1, A2, B2, A2, BN, and then AN, B1. AN, B2, AN, BN. So like this we are getting a compound. So in the case of conventional method, we are preparing only one compound at a time. So similarly many compounds we are preparing. So from that we have to find the most promising compound. We have to identify that. In the case of combinatorial chemistry or synthesis, we are getting different compounds simultaneously under the same reaction condition using the same reaction vessel in a systematic manner. So we can get many compounds simultaneously under same condition, same reaction vessel in a systematic manner. So we can get all possible combinations at once. So that collection is called as a combinatorial library. Then the library is then screened and the chemical compound, that is the active compound, would be finally identified. This chemical library or combinatorial library is having a central core structure, and that can be called as a scaffold, that is otherwise called as a common core structure or a common backbone. So what is it? Central core structure or common core structure or common backbone, and that can be called as a scaffold. Now we'll see the phases of combinatorial approach. So two phases are there. First we have to create the chemical library, then we have to identify the active ingredient. What is the chemical library? This is otherwise called as a compound library. So chemical library means that is collection of chemicals. What is the use of that chemicals? That is used in industrial manufacturing and also for high throughput screening. So each chemical that is having some information. So what are the informations available? One is chemical structure, physicochemical properties, purity and quantity. So these are the informations were available for the chemical in that chemical library. Now we'll see the types of combinatorial library. Two types are there. So first one is called as a scaffold based library. We have seen scaffold is central core structure, otherwise common core structure. So here this library is having compounds. That compounds are having a common core structure. So that may be single building blocks. I mean several building blocks. All are having similar scaffold, all are having a similar core structure, but the substituents may vary. So all the compounds are having the same scaffold or same core structure, but it can have many building blocks, single building blocks, and that is having similar scaffold. That is the core structure is common, but with the different substituents. Examples are amino acids, aminobenzophenone. So here this is aminobenzophenone that is connected with one more that is alkylating agent, another one is amino acid. So it is having a similar scaffold and that can have a single building block. So these are all the single building blocks. So many building blocks are there, and what is the common thing? All are having a similar scaffold. Next one is backbone based library. Here all are having a common backbone with the different substituents. Here building blocks are here in these case both were amino acids. So here what are the examples? That may be nucleic acids or proteins or carbohydrates. Here backbone is common one. Now we are going to see the techniques for creating the chemical library. Two techniques are there. One is mix and split method, another one is parallel synthesis. What are the two steps? Two methods. One is mix and split, another one is parallel synthesis. This mix and split is otherwise called as split and pool, otherwise mixing synthesis or portioning mixing synthesis. So what are the four names? Mix and split, split and pool, mixing synthesis, portioning mixing synthesis. So what is the use of this? By means of this we can produce large libraries of different compounds efficiently. So what is the process? How to create a library? First we have to divide a pool of compounds. Actually the collection of compound is called as a pool. That pool is having starting materials, many starting materials. So now we are dividing the pool of the starting materials into smaller portions. So that should be divided into smaller portions, and that portions are undergoing chemical transformation. After chemical transformation they should be combined. So which should be combined? The resulting final compound should be combined. So what is the method? The starting materials should be divided into smaller portions that should undergo chemical transformation. Then that should be combined. The resulting product should be combined. So by means of this we can get structurally different compounds. So 5 methods are there. One is splitting, reaction, mixing, iterative process, analysis and screening. So first is splitting. Name itself indicates splitting means that is the dividing. So the starting materials should be divided into equal portions, and that portions are having same set of compounds. So splitting is a division. So starting materials should be divided into smaller portions, and that should have same set of compounds. Next one is reaction. The divided portions are undergoing chemical transformation. So that involves that may be addition of reagents or functional group modification or other synthetic manipulation. Next one is mixing. So after chemical transformation, all are combined and pulled together. That is called as mixing. So what are the first three steps? Splitting, that is division. Then reaction, that is it can undergo chemical transformation. And next one is mixing, that should be recombined. Next is iterative process. So the first three processes, splitting, reaction and mixing is repeated many times. So several times that should be repeated. For each cycle we are introducing new variations or modifications to the compounds. So what is iterative process? We are repeating the first three steps. In all the steps we are introducing, all the cycles we are introducing new variations or new modifications to the compounds in the pool. Then that should be analyzed and screened. Why we have to analyze and screen? To identify the most promising candidate. So for this we are diagrammatically explaining this. First we are taking a bead, this is one compound. With this we are adding three compounds. So that is having two things. One is addition of two compounds with this. Here two compounds, here three compounds. So this bead is added with first this and this. So added with these two we got that. Then with the same bead, these two would be added, we got one compound. With the same bead, we are these three would be added when we are getting this compound. So that is having three substituents. So now collection of these, that is called as a pool. So with this pool, again we are adding two more compounds. So here 1, 2, 3. So three compounds we are adding, that is having two substituents, here 2, here 3. So already this pool is having these three, so that's why first compound has to react with these three and we are getting these products. So second compound, this has to react with these three and we are getting these three products. Then third, this is reacting with these three and we got these three products. We'll see that. So first this blue and number 3, that is reacting with this bead. So that bead is already having one blue. So two blues and 1 is there. So 1 plus 3. 1 and 3 would be joined. Again this reacts with this. So with the bead, two colors. One is blue, another one is orange. Then 2, 3. So we got that. Then this one with this. So bead with blue and green, and then already having 1, 2. So that is combined with the 3. So similarly we got these three products. So next one is called as a parallel synthesis. So from this we can generate large number of molecules simultaneously. That is parallelly we can get that, not sequentially. So we'll see the steps in parallel synthesis. First one is designing the library. How to design the library? Based on desired properties we have to design the library. So that library is having that may be potential drug candidate or a catalyst or a polymer or other materials. How the compounds are, that may be the drug candidate or a catalyst or a polymer or other materials. So how the compounds are? The compounds in the library are composed of different building blocks or different functional groups. So what is the first step? We have to design a library. On what basis we are designing the library? Based on desired properties. How the compounds are? That may be drug candidate or a drug catalyst or a polymer or other materials. So the compound is composed of different building blocks or different functional groups. Next one is a reaction setup. So we have to set up the reaction by means of the reaction vessel, or that may be using microplates or other formats. And that is having necessary, all the vessels are having necessary reactants. And next one is automation. So this is the main step in this synthesis. For this automation, we are going for robots or automated liquid handling systems for dispensing the reagents or mixing the reactions and the control reaction conditions. Next one is reaction monitoring. For this, we are going for spectroscopy, chromatography or mass spectrometry for monitoring the reaction. Then product isolation and purification. So after the completion of the reaction, the product is isolated and purified. So for this, we are using filtration or chromatography or crystallization techniques for purification. Next one is characterization. So after purification, we have to go for characterization. That is, we have to identify the compound. For this, we are going for NMR spectroscopy, mass spectrometry, and X-ray crystallography. Then biological testing or material testing. So this is the final step for testing the final compound for the desired activity. So two stages are there. One is a drug discovery or a material science. If it is a drug discovery, we have to check or we have to screen the compound for against the biological target, that is against the enzymes, to identify the potential drug candidate. In the case of material science, some properties would be calculated like conductivity, strength, and optical properties. They would be evaluated. So now we'll see the types or approaches of the combinatorial synthesis. So first one is the solid phase synthesis. Next is solution phase synthesis, parallel synthesis, mixed combinatorial synthesis, and other methods. What are the other methods? Multi-bin method, one bead, one compound method, iterative deconvolution, subtractive deconvolution, bogus coin detection, orthogonal peeling. So what are the main approaches? Solid phase synthesis, solution phase synthesis, parallel synthesis, and mixed combinatorial synthesis. So first is solid phase synthesis. Here we are using a starting material, and that starting material that should be linked with the solid support. What is the solid support used? That is the resin bead. So the starting material has to attach with the solid support. Then the starting material is undergoing many reactions. After all the reactions, the final structure is detached from the solid support. So now we'll see the essential requirements. So one is the solid support, another one is the linker or anchor. How the solid support is? That support is a cross-linked, insoluble, and polymeric support. How the support is? That should be cross-linked, that is insoluble, and is a polymeric one. And how that is? That is inert to the synthetic conditions. Example is a resin bead. Next is an anchor or linker. That is linked to the resin. That is linked to the solid support. How that is linked? By means of covalent linkage. Now this anchor or linker is having a reactive functional group, and that is used to attach the substrate, and this has to attach with the substrate. And now there was a bond that is linking the substrate to the linker. So how the substrate is connected with the linker? By means of a bond, and that is stable to the reaction condition. Then after the process is over, then there is a means of cleaving the product or intermediate from the linker. So main 2 points is we need a solid support, we need a linker. Solid support is so the solid support, it is having the essential requirement as solid support, a linker or an anchor. So solid support is inert to the synthetic condition, whereas this anchor is having a reactive functional group that can attach with the substrate, and there was a bond that is linking the substrate to the linker, and that was stable to the reaction condition. And finally, after the process is over, then there was a means of cleaving the product or intermediate from the linker. So we'll see the solid support. So first successful example for this solid phase synthesis is Merrifield resin peptide synthesis. So here what is the resin used? That is having polystyrene bead, where the styrene is partially cross-linked with 1% divinyl benzene. Then the beads are derivatized with the chloromethyl group, that is the anchor or linker with the amino acid that can be coupled through the ester group. So what is the anchor used? That is the chloromethyl group. And what is the resin used? That is polystyrene bead. Here the ester is stable to the reaction condition, and that can be cleaved at the end of the synthesis using vigorous acidic condition. What is the acid used? That is called as a hydrofluoric acid. So in the case of Merrifield resin peptide synthesis, what is the resin used? Polystyrene bead. And what is the linker used? That is the chloromethyl group. And for cleaving that, we are using acidic condition, that is the acid used is hydrofluoric acid. Example for this are Tentagel, Pepsin, and Pega. Tentagel is polystyrene polyoxyethylene graft copolymer, and Pepsin is polyamide support that is mainly developed for peptide synthesis, and Pega is polyacrylic amide ethylene glycol copolymer. So what are the examples? Tentagel, Pepsin, and Pega. So next one is anchor or linker. Already we have known that is covalently linked to the polymer chain for making up the solid support. So that is having reactive functional group in which the starting material can react and that can attach to the resin. So resulting link must be stable to the reaction condition throughout the reaction condition, but that can be cleaved to release the final compound. So finally that would be cleaved, that should be broken. So here the linkers are distributed in the polymer chain, but most of them are in the interior of the polymer bead. So where they are in the interior position of the polymer bead. So different linkers may be used depending on the functional group with which which should be present on the starting material. So based on the functional group present in the starting material or the functional group that is desired on the final product once it is released. So based on these, the linkers should be classified. So example for the linkers are Wang resin, Barlow's resin, Rink resin, dihydropyran derivatized resin. So first one is a Wang resin. So it is having linkers that are suitable for the attachment and release of carboxylic acids. So what is Wang resin? That is having linkers that are suitable for the carboxylic acids, attachment and release of carboxylic acids. It is mainly used in peptide synthesis by linking an N-protected amino acid to the resin with the help of ester link. So this is stable to coupling and deprotection steps in final protein synthesis. So finally it can be cleaved by using acidic condition. What is the acid used? Trifluoroacetic acid. And finally the final peptide is released from the bead. So what is the main limitation for this? So first amino acid is having chances of racemization, that is prone to racemization. So this is an example for Wang resin. So this is mainly specific for carboxylic acids. So next one is called as the Barlow's resin. This is having a trityl group, so that is acting as a linker. So that trityl linker is designed to avoid the problem of racemization. So the final product is cleaved under very mild conditions. So what are the examples used? Acetic acid, tetrafluoroethylene or dichloromethane or trifluoroacetic acid, owing to the high stability of the trityl cations that are formed. So here trityl cations are formed that are highly stable. That's why we are using these examples. So the molecules can also be linked to the resin with the help of an alcohol group. So this is an example for Barlow's resin. So next one is the Rink resin. This is suitable for the attachment of carboxylic acid and for the release of the carboxamide. Here the starting materials with the carboxylic acid can be linked to the Rink resin by means of an amide link. So what is the link here? That is an amide link. So after the reaction is over, it is treated with the TFA to release the product with the primary amide group, not getting the original carboxylic acid. So final product is having a primary amide group. So this is an example for Rink resin. The rounded portion that is going to add. So last one is a dihydropyran derivative of resin. This is mainly suitable for alcohol. So primary or secondary alcohols are linked to this resin. What it is? That is dihydropyran derivative of resin. So alcohols are linked in the presence of pyridinium 4-toluene sulfonate in dichloromethane. So after the reaction is over, then that should be cleared by using TFA. So this is a structure of dihydropyran derivative of resin. So that's all about combinatorial synthesis. Thank you.